Well, I realize how fast this time goes when you have so many good things to talk about. So we're going to go ahead and dive in. Maybe I'm going to introduce. You know what we'll do is I'm going to, because of time, I'll have you guys introduced when you do your introduce yourself when we do your thing you're excited by. So my name is Mike Boyle. I hope to lead the science. This is part of our clinical science team. We don't have all that long, and so what I wanted to do, and we're going to make sure that we had a chance.
So Mike's going to talk faster this time.
No, exactly, really fast. We want to make this interactive and be able to answer questions. So what I was going to do is maybe give about a 10-minute overview. I am going to talk pretty fast because there's a lot there. And then have each of the members of our team introduce themselves and maybe talk about one thing that they're particularly excited by. And then we'll throw it open for questions. So I think one of the things that may be worthwhile just thinking about is, because you hear about all these different projects going on, I just thought I would tell you about how we think about it and sort of organize some of the research at the foundation. From the more basic science research into new therapies, all the way up into things where there's approved medicines and we're trying to figure out how to help people make sure they take them. And we're going to focus on this session really on what our team works on, which is this red box, which are really new therapies which are in trials in people. So this is not preclinical where we're just testing in the lab, but they've been advanced enough to go into that. And we really divide those into 3 buckets. One would be basically drug company studies, phase 2 and phase 3 studies where we're saying, hey, is this going to actually work in people? The other one would be actually projects that the foundation does. Sometimes they're drug studies, but other times they're aspects of care that we're looking at, such as how long should we treat people with IV antibiotics, those type of things. That's usually for trials that we think nobody's going to do this for a drug company. We need to do this as a foundation because we need the answer, and then no one's going to make money from this. So we want to actually make sure we get the answer ourselves. And then lastly is a group where we ask all the experts in the field, investigators from hospitals and medical centers around the country to propose ideas, and we fund those, particularly the ones that we think have the most potential to make a difference. And that I think one of the other things that's a key message here is there are a bunch of clinical trials going on. As much as we tend to hear mainly about the modulators, there's a whole bunch. As a matter of fact, there's more new clinical trials that are going to be starting this year that are non-modulator studies than are the Restore CFTR studies, or the modulators. That I will just mention at a fairly high level a couple of the big areas, and then we'll dive in. So the modulators, we're all familiar with the Vertex trials. There's 2 different compounds right now which are moving forward. 659 is one of them, which is the one that's starting right now. This is part of that triple. That's going to start in people with a single F5 weight del mutation, and then in the next month or 2, move over to people with 2 F5 weight del mutations. That's the one that right now we really think has a chance to move the needle. There's other companies, though, that also have triple. They're earlier. We haven't seen any clinical results from these triple like we have with Vertex, which looks so promising. But Galapagos AbbVie and Proteostasis both have plans to move forward a triple this year. There's other companies such as the Flatley Lab, which is also doing some corrector and potentiator studies, and then 2 other companies that have very promising modulators in the lab that we're hoping to have move into the clinic in the next year or so. I'll just mention this quickly, that one of the other exciting things, and actually, Tony will talk a little bit about this, is there's some new opportunities for using the lab to be able to identify who might be able to benefit from these new modulators, particularly with rare mutations. And that the other thing that you'll see as a theme this upcoming year is streamlined studies, particularly for modulators, because I think there was a question in yesterday's session was, how can we do studies when there's existing therapies, particularly for modulators, and that many of the newer studies are going to be a little shorter and sweeter because of existing therapies, and we know we can't use long placebo. This is actually the phase 3 design for the trial that's just now starting. This is the triple. And the bottom line is that because there's no existing therapies, there is a plan for 6 months placebo, 6 months treatment, just like we normally see. But this is really interesting. The primary endpoint, the key measurement, is at 4 weeks. That's never been done before, where you say after 1 month of treatment, they're going to look and see, is this working? And if it does work as well as we've seen so far, that's going to open up a whole bunch of opportunities for thinking about, are there ways to be able to get drug to people who are particularly sick or move faster in approval, all those type of things. But that is lightning fast, and that's a credit to the FDA. And then I'll just mention, for those people who are in that 5 to 7% group, there's a trial that's going to be moving forward. This is really early, but the whole idea would be to develop a method to deliver the correct copy of the gene, or at least in this case RNA, that tells the cell how to make the cystic fibrosis protein. So it doesn't matter what mutation you have, because if you have that corrected copy. And so Translate Bio has an ability to put RNA into the middle of this nanosphere, which basically means a very small sphere, which can be inhaled into the lungs, can get into cells, and allow those cells to be able to make protein. Now, this is early, like I said. It looked promising, has looked promising in animals, but this is the one that we're saying, hey, this is at least something to be looking at for that 5 to 7% group. We mentioned anti-inflammatories as being something. This is a big year for anti-inflammatories. There's 3 different trials. Celtaxis is actually, and there should be a T in there, but Celtaxis right here, they have a large trial going on that will read out this summer. And we'll actually find out whether or not their anti-inflammatory is actually going to be able to make a difference. That's another one. It doesn't matter what mutation you have. CORB has had promising phase 2 results. They're going to be starting a pivotal trial later on this year. And then Laurent has an ongoing trial a little bit earlier. A lot of that's being done in Canada, but some in the United States. And then Poly4 will be later on this year. And so in the next 2 years, we'll probably have a much better understanding of whether or not some of these new therapies that treat inflammation are going to be helpful or not. And then, of course, there's anti-infectives. So just to highlight a couple of these things, because people tend to think about what's the bacteria they treat. For people with MRSA, there's this ongoing study of inhaled vancomycin. This is basically like dry powder Toby, but it's dry powder vancomycin. And that would be something that would give a chronic option to treat people with chronic MRSA, which is about 25% of people with cystic fibrosis now have MRSA. And so there's another drug, tobramycin with fosfomycin added, which helps to treat both the Pseudomonas better and also MRSA. There's other drugs that we've heard about before, which are ongoing right now, trials, gallium, nitric oxide. These have opportunity to treat not only bacteria, but other things such as NTM, fungus. Actually, they tend to be broad spectrum. And then I'll just mention a couple other ones. This is a very potent but early antibiotic that's being tested in Belgium. We're paying for all that work. And then itraconazole is an antifungal that's being tested. We're helping to fund this, even though it's actually, the trial is not in CF right now. It's actually being done outside of CF because it's, but anyway, this is something that could potentially benefit people with CF in the future. Mention 1 or 2 fun ones. This is a Niklaprim. The reason I mention this is because this is one that actually could be at a drugstore near you sooner than a lot of these other ones, because it's actually already had a lot of success in other diseases. Particularly, this company has completed a trial looking at skin and soft tissue infections. And so it's been shown to be effective there. It's now in a pneumonia study for a non-CF. But the reason we're excited is because this drug has potential to have effectiveness against Cepacia, against MRSA, against Achromobacter, against Stenotrophomonas. These are ones that are sort of our FBI most wanted list for bad bacteria. And so this would be low-hanging fruit. We've actually are paying right now for them to test in John LaPuma's lab in Michigan this against a bunch of all of our CF samples to see how effective it is. The first part looked great. This would be very easy to plug in, right, into a trial in CF because it's already been shown to be safe and effective in other diseases. Somebody asked about Pseudomonas in the last section. This is just another, and actually Tony mentioned about problems with biofilm. This is a drug which a compound which takes regular tobramycin but adds a compound, calcium EDTA, which helps to break up some of that biofilm. And in a study in Australia actually had pretty significant effect on FEV1. Now, I say be careful about these numbers because they're big, and it was in the setting of exacerbation, so the numbers are big. But they're going to be doing a trial that we're going to be funding to see whether or not adding calcium EDTA can help tobramycin. We're always interested in better mucociliary clearance. Alagoji is one of these drugs which breaks up biofilm really well in the dish. The phase 2 study didn't look quite as good as we were hoping, so we're adjusting the dose. This is being done in Europe, but this might be something that could help with breaking up mucus. And then there's 3 different studies going right now looking at ENaC inhibitors. Remember, ENaC is a sodium channel which lines the lungs as well. It's actually overactive in cystic fibrosis, and that helps to dry out or causes the lungs to be dried out in CF. So that inhibiting ENaC inhibitors would actually add more liquid to the lungs. We're going to see. This is something that lots of people are convinced is going to work. It works well in animals, but we actually haven't seen it really work in people yet. So we'll know a lot more in this next 2 years. And then the last thing I'll mention just in terms of budget is that, you know, the money that people raise in all these events and work so hard to, it doesn't just go to those trials and to enable drug companies to move forward. We want to help them go faster. We want to draw in the best things into cystic fibrosis. But a lot of this money goes into building our network. So about $25 million a year goes into the research teams, those 89 centers around the country that really focus on CF clinical trials. And so we help to pay for those teams. There's also grants, and Dara's going to talk a little bit about this, but grants where we ask for the best ideas. Anybody who has an idea that we think can make a difference, we spend about $25 million a year supporting that. And there's also a couple of specific clinical trials, as I mentioned before, which I'll highlight here, which we say we need to do as a foundation, because no drug company is going to do them. And some of those, so that gets us to over $50 million a year just in beyond the typical things that you think about in terms of the drug company support. And I'll just give you a highlight of some of this list of all the other things going on that the CF Foundation is basically doing by itself, because we know we need to. And so 2 of these are looking at the long-term effects of ivacaftor or CAMB, trying to see how it affects other organs, how it affects diabetes, how it affects the sinuses, how it affects GI. Looking at gallium, which I mentioned before. This is one of my favorite studies. This is going to change the way we treat exacerbations. So without going into a whole bunch of detail, there's one group, basically after about a week, 5 to 7 days of starting IV antibiotics, people do a pulmonary function test. If they are already improving, they get randomized to either 10 or 14 days. If they're not improving, they get randomized to 14 or 21 days. And this is going to tell us of whether or not if somebody starts to respond quickly on IV antibiotics, if we could treat them for only 10 days, get them out of the hospital earlier, have them do less time missing school. Also, is there a benefit to treating longer for people who aren't? So this is going to change the way we approach. And then SHIP, hypertonic saline, we're going to see how well it works in young kids. We're also looking at glutathione, not for lungs, because that's sort of questionable how much it works there, but nutrition, as well as infant nutrition, a couple other ones which you can ask me about afterwards if you need to. Everybody's asking about NTM. Be aware of the NTM Consortium. This is a group of sites that we're funding specifically to focus on developing the best care practices, as well as sort of the network to be able to do research for the new therapies that you heard about yesterday from Dr. Skach. And then one other neat one, because I think part of this breakout session is to be able to take home a couple of pearls where you can say, oh, there's something neat going on, is this one that's we call Check SC, which is really, it's going to help us understand the role of sweat chloride in monitoring how well someone's responding to modulators. We know that when modulators work well, their sweat chloride comes down. We've seen that with really effective things like Kalydeco. So one of the questions we have is, over time in the future, could we use this as a way to assess and sort of monitor people, almost like a blood pressure, to say how well is their cystic fibrosis protein working? So maybe that every time you or your child or your loved one or whatever comes to clinic, we might actually check a sweat chloride, or maybe something you would actually check at home from time to time with some new technology will help to sort of see how's this going, how am I doing. Anyway, we need one step at a time. I'm sort of talking about way down the road, but one step at a time. The first of all is to say, let's figure out that relationship. So that study is going to be looking at that over time. And then GI, a big topic. I'm going to skip it because Dara's going to talk about it. And I'm going to skip through this. Just I will mention one thing which I didn't mention last time. That is, as much as we focus on the U.S., we realize that this is an international problem. So we put about $25 million into our network in the U.S., but we've put a couple of $1 million actually to outside the U.S. because we know that some of these trials actually work best in Europe. We're actually supporting the European network. We're supporting the UK network. We're actually helping in Canada now, Canada and Australia as well, because we want to be able to go as fast as we can. And so literally the last point I would just make is that this requires teamwork. You know, we can only do this. It's exciting to have these leads. It's exciting to have this promise. But this really requires the team of people with cystic fibrosis and their families participating in the trials. That's the only way we're going to figure these out and figure it out as fast as possible. So you'll see this on our website. We feature this. We talk about it with videos. We feature people who participate. You'll see it on social media. This is part of the social media campaign. But it just highlights how we're all in this together. It couldn't be a more exciting time, right? It's great to have these, and so good things ahead. So with that, I wanted to introduce the rest of our team, or have them maybe introduce themselves, because this is the team that sort of covers that a bunch of that clinical trial space. And I asked each of them to not only introduce themselves, but to talk about one thing or so that they're particularly excited by that's ongoing, and then we'll go to questions and answers. So Dara, do you want to start off?
Yeah. I'll start off. We could have a whole session on what's Dara excited about, but I'm just going to choose one, which is the GI study. It's such a huge issue for people with CF. In fact, about 6 months ago, I did a survey with the community and asked them to rank research topic areas, and GI issues came in number 2. That was 2,000 people, almost 2,000 people that responded. Number 2. Priority research in GI right after infection. And just to echo that, yesterday when Danny was sharing his story about his transplant, the one symptom, the one complication that almost broke the camel's back was a GI symptom. This is very important, and we're starting to tackle it, which is very exciting. So it's very early in the beginning planning process of learning more about GI symptoms. And the first step is really to identify a way to measure GI symptoms. And if you think about it, when you have a lung clinical trial that involves pulmonary problems, we have FEV1 that measures if things are getting better. So you can see an improvement in FEV1, and that's how we know it's working. We don't have that measurement for GI symptoms. And so that's our first step, and we're going to need the community's help with this, because first we need to figure out, are we looking at the right symptoms, and are we able to measure it correctly? So if you haven't done so, go and join Community Voice, because we need your help with this. So that's what I'm really excited about. I'll stop there because we don't have a lot of time, and I want to answer questions.
Actually, you want to give 1 or 2 lines about you and what you do?
Oh, yeah. Oh. Who are you? I'm so excited that I forgot to mention who I am. So my name is Dara, and I started the foundation about a year ago, a little bit more than a year ago, and I oversee the investigator-initiated clinical research studies, and that's a really big way of saying your doctors, your clinicians, and your scientists at your institutes, when they have those really great ideas, and they say, I think this could help somebody with CF, and they need some funding to help make that happen, then they come and submit grants to my department, and we evaluate whether it's worthy, if it's going to move us towards the mission, if it's going to help people with CF. So that is what I do, and I also have 2 children with CF. I have an 8-year-old Maximilian and a 6-year-old Penelope, and they're doing great.
Thank you. Thank you, Dara. I'm Patty Burks, and I'm a nurse. I was a research coordinator at Washington University School of Medicine, doing CF research exclusively for 9 years. 2.5 years ago, I came and joined the foundation and live in Bethesda, where I help support the 89 research teams across the country that do our CF clinical drug trials. What am I most excited about? Well, I'm pretty excited about the next generation modulator studies, so that's pretty cool. But maybe I'm more excited about the network that does those trials. They're having their 20th anniversary this spring. The TDN, Therapeutics Development Network, has been around 20 years this spring. That's pretty amazing. If you look at the pipeline, that's what they've been doing with the last 20 years. But maybe I'm most excited about the patients and families who continue to enroll and participate in these trials and moving the ball forward. So if you are here and your loved one ever participated in a clinical trial, I'm most excited about you. Thank you.
Hi. Hi, I'm Tony Dermowitz, and I'm a pediatric pulmonary and critical care physician who spent a long time in academic medicine, mostly at the University of Colorado, doing a lot of research on lung development predominantly and clinical care. Then I spent a short period of time in the drug industry helping develop monoclonal antibodies with a company called MedImmune, which is best known probably in pediatrics for their anti-RSV prophylaxis monoclonal antibody for babies. For the past 12 years or so, I have been working in the Division of Pulmonary Allergy and Rheumatology at the Food and Drug Administration, helping review drug products and decide what kind of action to take on applications. So I'm happy to be at the CF Foundation now. I just wanted to say one thing real quick. I need to leave pretty much right on time for other obligations that I can't get out of. And I don't know if my name is on the website because I've only been at the CFF for about a month and a half or so, but it's like the A. Dermowitz, and it's like the @cff.org. And if you have a question that you think I could answer, you can either email me or email me. We can get a phone call together because some of these questions are not easily done, and I don't type very well.
Okay.
You want to talk about what you were excited? One thing you were
Oh, the excitement part. Well, you know, I think it was very exciting in a first step to have data that you could develop and look at in a laboratory help support access to certain modulator drugs, mostly Kalydeco and now Symdeko. But I think what I'd like to try to do and try to see if we could make it happen is to be able to help develop a universal lab test that could be able to support drug approval across different companies. Right now, the only lab test that's been validated, if you will, is one that Vertex has. But there's, as Mike said, there's a lot of other companies that are trying to develop drugs. And they otherwise would have to have their own individual tests and each one go through the machinations each time. But if we could get together and it's kind of like herding cats because there's a bunch of people that have their own test, figure out which one we can move forward with and maybe act as a broker for that, both to help approve drugs and also to help individualize therapy later on when they're on their market. That's something that I think is on the top list of Tony Dermowitz's things to work on.
That's great. And we actually, I'm going to brag about the team a little. We just have such a great team, both on the science side, but also just the passion part. We have some direct CF connections. People have worked, Tony's soft selling himself as usual, but has been just a thought leader for approval for all the drugs that are currently on the market for modulators, and has led the whole approach to using the lab to expand the number of mutations that are eligible. So we're excited to have him on our team now. So what I wanted to do is to make sure we had opportunity to answer some questions. I mean, if we need to, we can. So I think we're going to send Patty into the audience. Dara has online, so if you're online, feel free to submit. Is there anything hot off the presses there or not?
Nothing hot off the presses.
Is there any?
I think there's a question
Okay,
right
perfect.
over here.
All right, so this is easy.
I don't know who. I'm sorry. Who is that?
That's right.
Hi. I have a question about the triple study that just started. I noticed the placebo at the bottom, but if you've already been on a modulator, how are they going to do that with a placebo?
Yeah, well, so actually, let me go back to that because actually I probably didn't explain it very well because the placebo actually is, let's see. So it depends on the study. Well, I'll just explain it. Let's see. So there are going to be two different designs. Now, for people who have just a single F508 del and don't have, don't have any mutations that are eligible for modulators right now, those people won't have been on modulators, so they can go on placebo, right? So if you have a single and your other mutation is, so basically the people that are eligible for this trial have a single F508 del and another mutation that doesn't make any protein. They don't have any modulators right now. So that group can use placebo. They don't have anything. It's a different design for the people with two mutations. They're going to be compared to Symdeko. So the group, when that group will have one group that will get the triple and another group that will get Symdeko. Does that make sense? And then that way you can actually see the difference. And that's just, this is a key thing. When you see these data, actually I'm going to say this right now just so you remember this. When you see these data coming out with the triple, look and see whether or not it's reporting a single F508del or the double. The single's always reported against placebo. So when you see that 13% improvement, that's compared to people who are taking placebo. And that's because they don't have existing therapies. But when you see those numbers for the double and they say it improved 10%, you say, that doesn't make sense. Why is it smaller? It's because it's being on top of what their Symdeko that they're on, right? So does that make sense? So basically, if you don't have therapy, you're going to use placebo. If you're on Symdeko, they're going to have you stay on that. Well, if you're a single on Symdeko, that's probably, you have to follow the rules for this study, truthfully. That's something you have to talk about with your care team. But I can tell you, here's the thing. They're going to want to, anyway, they are specifically designing this trial to treat a single F508del, another one that makes no mutation, and compare to placebo. The one good thing about this, as Tony sort of mentioned in our last session, is the readout's going to be quick. And then there's going to be some opportunities to... actually, Tony, I thought it was an important point last time. Do you want to talk a little bit about sort of what you picture as this starts to play out if there's good results?
Where's it?
As Mike was pointing out, if you look, the main endpoint of the study for approval, if you will, is after a month. It's not at the end of the six-month trial. So, if those results are positive, and it takes a while to figure out whether they're positive because you have to get all the data in, all the stuff, all the statistical people have to jump all over it and things like that. If that is positive, then the company could ask to make the drug more available to people and have an early access program, if you will. And that would make the drug, like I said, more available for people because they know it already works. They just have to get through the paperwork and through the review and all that kind of stuff to do that. So that's a possibility that Vertex could do. I think they have to kind of want to do it, though. And so that's a discussion they'll have with the FDA at that time.
So I just think one of these things is it's moving so fast right now. Sort of buckle your seatbelts. The first thing we need is to be able to get the results, but then it'll be interesting to see, particularly for some of the access programs. So it looks like we have plenty of hands. Why don't we?
Yeah, I just want, while you're getting to the question, I do want to clarify something because we talked about this at the last session. So the triple, even though the clinical trial has a single F508del and then a list of mutations for the trial, but the hope is that once it's approved, it doesn't matter what that second mutation is. As long as you have one F508del, then you will be eligible to take that. But it's the trial where you have that limited. So don't think that if you have that second mutation, it's not going to work for you after it's approved.
Thank you, Dara. I have a question right here from Cheryl.
That may be the most important point today.
Yes,
That's right.
Thank you.
Yes, I just had a question about the NTM consortiums. What will that encompass, and then what is kind of the expected outcome or hopeful outcome in creating those?
So I went through this in about 10 seconds, and truthfully, there's a load of work going on this. So there's two current studies going on called PROMISE and PREDICT. That's right, PREDICT. Thank you. Too many P's. There's another one. PREDICT. That initially it is to establish sort of this best standard of care. So right now, if you know, it's very complicated to treat a lot of NTM, whether it's MAC or abscessus, but particularly abscessus. And so we paid for a bunch of all the best NTM doctors to get together and to say, what do we think the best treatment approach should be? And then to actually get all these centers to implement it to see, okay, here's what the best you can do with the current therapies. It's almost a baseline. The nice thing is, at the same time, we've got several companies, 4 different companies that are actually proposing new therapies for NTM. You heard about them yesterday. Bedaquilin's one of them. Inhaled GM-CSF is another one. Gallium's another one. Inhaled NO is another one. The idea then is actually this year to be able to use this consortium to be able to add on some of these to start to say, does this get us better results than what the standard we've been studying? So the end point of this is not just a single clinical trial. This is almost an ongoing way to learn the best way to treat NTM. So you start off with saying, okay, we're going to standardize care. We've published some guidelines. This is the way we're doing this. This is what it results in. Now we're going to add in some of these other therapies and try to basically come up with something that works better than what we have, particularly for abscesses. So does that answer your question? Yeah.
That's great. And now Angela has a question.
Hey, Mike.
Hey, Angela.
Hey, did you say yesterday, did I hear you say that Symdeko works a little better with itraconazole than our CAMB? And if so, how would you decide, or how would the doc decide how to moderate the dose for Aspergillus?
Tony, you want to take that one, antifungal? You want me to do it?
Let me see if I have the question.
I think it's basically Symdeko's interactions with antifungals.
Symdeko doesn't have nearly the same type of interaction or the amount of interaction with antifungals as Orkambi does. So it makes dosing it and adjustments and if you look at the label, contraindications to using it from a practical standpoint, a lot easier. So if you do have or are taking a drug that has a lot of interactions with Orkambi, that could be, and you need to take that drug. That could be a rationale for getting switched over.
Other questions? Dara, do you have any?
Actually, I was just say one last thing, Angela. As Tony mentioned, the label is actually really helpful. So you still need to tinker with the dose a little bit. With the differences with our CAMB, you don't get any levels for itraconazole. It means we can't take them together; it doesn't work. But with the Symdeko, as Tony was saying, you can. But you look at the label, and you can actually give specific guidelines depending on the antifungal you use, how to adjust the dose.
That's great.
But it opens up a whole new world for a bunch of people who were taking antifungals before that couldn't take it. Yeah. Any other? Oh, we got Doug.
Here you go.
Doug knows all the answers. You're not allowed to ask questions.
Here you go.
Well, I'm your resident naysayer, as you know. You're talking to a very motivated, eager group who are just trying on the edge in the cup of something new. Mm-hmm. And you have staring at you the Kaleido, yellow light blinking, a home run. This is a grand slam. This really is a terrific product, and we can't get past 60% adherence. And I would say to you that almost as equal importance as all of these wonderful new cutting edge things is work on behavioral modifications, like how is it that when you have something really terrific, the human nature says, I don't want to change, or I'm afraid of something new or different, or it's too much trouble, or I don't trust what you're telling me. And I'm just pleading with you that you can put bazillions of dollars into great cutting edge. And if anybody wants a mRNA modifier, a regulator more than I, but we're missing something when you have excellent material and the great percentage of the ordinary clinic population isn't taking it.
Well, I might argue with you a little bit about the numbers, but I think I totally agree with you that that's an important topic. We didn't talk about it today mainly because it's not what our team does, but we have a whole team that's what they do. So this is actually this whole group of centers called the STRC, which is a success with therapies, gets that exact point which you're talking about, which is we want to help people to be successful in actually taking the things we know works. And that's in one way, the pills are easy, right? What about if we know they need to be taking inhaled antibiotics and airway clearance, and it gets so complicated? So couldn't agree more that this is a really important topic. Cindy George, who many of you may know, is actually leading this effort for us. And we have about 25 centers who are funded specifically to do nothing but adherence research. Actually, we've gotten away from the word adherence a little bit. It's supposed to be sort of partnership, success with therapies, and to do exactly what you're saying. How can we actually help people to be successful with doing them? There's actually some neat studies that are ongoing, some telecoaching, some telehealth, some apps, some other things for being able to dive in on this. The mental health effort was as well, because a lot of times depression can play a role in this. And so, you know, the foundation has funded a large number, as you know, of mental health support people at centers to try to get this exact point. So anyway, Doug, I agree with you. Hopefully we'll continue to make just as much progress and as much attention in that area as for the new therapies. But I would like to have the problem of having the therapies and then having to learn to take them. That would be my goal. So was there another question over here? I think, Patty, I think there's one right here.
Oh, there you go.
Tony, you need to go? I know you've got a family event. Okay.
So my two boys have been on our CAMB and have gotten great results, and they will be transitioning to Symdeko in the next week or two. But my question is, for them, for me, I'm the mother, I'm so happy and grateful. For them, when we started on Orkambi, they're like, awesome, it's 4 more pills I have to take. So my question is, the sweat chloride thing, potential, could that potentially be a way to gauge whether or not they could start backing off on some of their treatments? Like, this seems arbitrary now. Like, they're doing awesome, but how do you decide, like, when you can do? I know some people have stopped, you know, they just don't do hypertonic anymore or whatever.
Yeah.
For them, that's the important thing. That's, if they don't have to do so much every day, that's what the point they want to get to.
So can you pull out? So I'm happy to answer that question. We've talked about this a lot, and that's actually a very common question. Do you want to take? You want to take a look? I can grab it. You want me to grab it? Okay. Okay. So I think there's 2 things to this. One is we would like to be able to study this as well to figure out, okay, is there a certain point we can actually do less? That's really the goal, right? I do want to caution a little bit that I think the people that will really have the greatest ability to do that are people with more mild disease. Right, so if you truthfully have sort of mild disease, you're doing some things to try to prevent problems, I could picture that really being the case. I think it's a little different story for a lot of the adults that I take care of who already have a lot of structural lung disease. They're still going to have areas that collect mucus. They're still going to have areas where there's infection, right? Because that's going to get better, but it's not going to go away. So I think there may be some opportunities to be able to cut down some, but I actually have really been trying to caution some of our adults with sort of not having too much, or not promising too much, because we know that, for instance, there's other patients that we'll see at times that have just regular bronchiectasis. It's not cystic fibrosis, and they actually need to take treatment, even though their cystic fibrosis protein works well, and that's because they collect mucus, so an airway clearance. So I think a lot of it's going to depend on where you are when you start and how much damage you have. But to get to your answer, we are actually going to be funding in this upcoming next year or two some studies that may look at could we actually remove something in this setting. We were trying to think about how to do it. It's complicated. We want to make sure we do it without hurting anybody. We may be able to use the registry and be able to study people who are doing it anyway and seeing if it makes a difference. So I totally get it. I think we want to go there. I also probably want to just be a little bit cautious, so we're not trying to promise that if you have that even with the triple, I think people are still going to they're going to be better, they're going to be sick less, their lung function is going to stabilize. But if you have a lot of scarring, you're probably still going to need to do some things to clear mucus and keep infection in check. At least that's what we're thinking. And then eventually we'll get to the day where people start so early we don't have to worry about this at all. So, yeah.
I just want to add to that. So the survey that I spoke about earlier where GI was number 2, reducing treatment burden was number 3. And what's interesting is in the beginning of the year, we had the Clinical Research Advisory Board, which brings in experts, our clinicians, and there was actually a discussion about how that's a priority for them as well. They recognize that it is important for us to do that. And I think in the future we're going to look at the next couple years as before the triple and after the triple, and I think it's going to change the landscape of our research and looking more at how we treat infection and how we treat inflammation and things like that with the triple. So it's in our minds. It's just like Mike said, we have to be very cautious and we have to do it very smart.
Yeah, and actually at the North American meetings this year, one of the things we're going to be doing is actually have a session specifically for the plenary about infection, but say future of treating an infection in the presence of the triple, because it sort of changes the way you think about things. Same with mucociliary clearance, same with inflammation. So those are all incredibly important topics. We know we continue to need them, but we want to think about, okay, how's the landscape changed? Maybe a last question or two. Dr. Dermowitz, have a wonderful time tonight with your family. You know what I mean? So as Tony volunteered, I think that if you were having specific theratyping or rare mutation questions, you could send him. Yeah, that'd be perfect. Thank you. Safe travels. So I think if anybody wants to go. If you have questions, we're happy to take some more, because I think we're officially done at 5, but people are just going back to their rooms or whatever right now anyway, right? So feel free to go if you want to, but we're happy to stay. So was there a question over here?
Yeah. I can just come up there. Okay.
Well,
My question
ahead.
was on the ENAC, where you guys were looking over at the ENAC system. Is that going to be given to everybody, or I feel like the research into ENAC's pretty new. Are we still trying to identify who has an ENAC exacerbation to their disease and they're going to get that therapy?
That's a great question. I think one of the things that's most attractive about ENAC is it could benefit everybody. So it doesn't matter what mutation you have. And we don't, it's a good question is there some way that ENAC is particularly acting up and we need to treat this group? We don't think that's the case. We think it's acting up in everybody, and that's contributing just to the typical problem. So the reason it's sort of intriguing is that there's an animal model that looks a lot like cystic fibrosis, where when you make ENAC overactive, and that when you actually treat that ENAC overactivity, those animals look a lot better. The thing that hasn't happened yet is to see this in people. So there's a bunch of companies. Vertex actually had an ENAC inhibitor that didn't work. So they did a trial, 371. They went through that. It didn't work. There was some question about whether or not it was that good a drug. I think everybody still thinks this is maybe a good target because it can benefit anybody. So I would say, let's see how these next 2 or 3 companies do. If 2 or 3 companies all block it in different ways and patients don't get better, then we probably need to look at a different target. But it's promising, so we're hoping. So, any other questions?
Oops, sorry, there you go.
One quick question, and a patient had actually asked me, for the rare study, she's been transplanted, but she definitely is homozygous.
Yeah.
So she was like, but what does that have to do with anything?
Yeah. So just to mention, the thing called the RARE study is actually, we mentioned this yesterday when we were talking about how we need cells from people that have 2 stop mutations. Because we want to be able to test new therapies. And the problem is if you're trying to test them and the cells have 1 stop mutation and then F508del or some other mutation, whenever you see a response, you can't really tell. Are we actually helping the stop mutation? Are we somehow affecting this other mutation? So people with 2 stop mutations, which is only probably 1% or 2% of people with cystic fibrosis, maybe 3%, that's very valuable. I think transplant doesn't matter, truthfully. I think I'd have to look at the exact...
So transplant's listed as exclusionary criteria for the RARE study.
Okay, well that's interesting.
Yeah, it is.
That is a very interesting one. Yeah, so that we always tend to exclude in CF studies transplant because we feel like, okay, that's a sort of a separate issue. I think it's a really legitimate question because you're taking nasal cells.
Right.
I mean, the one thing there is that they're on a lot of immunosuppressants, but I think if I were actually rewriting, I didn't write those, but if I was on a team that was rewriting those, I probably would say, why don't we take anybody?
Right. And who's the physician over that study? Is it?
It's Steve Rowe.
Steve Rowe. One could just send him an email.
Yeah, I mean, they'd have to actually go to the IRB and get a request an exemption.
Yeah, it said like futuristically things may change and they may need more in their database at some point. But she's like, I'm post-transplant. I can't do this and I can't do this. This is something I could possibly help with.
Oh, that's very kind.
So,
yes. Well, you know what? I think it's worth asking Steve and the team. We'd love to have those cells. And I agree with you that lung transplant doesn't really affect the nose. So,
Not as far as I know.
Sue Hook. Oh, was there another one in the back?
Sorry.
Yeah, well, maybe we'll do 2 more questions, and then anybody else who wants them will just come up. That way people can feel free to. Sue had a question up here.
So my question is the percentage of our patient population that have nonsense, and then are you just saying that 2 to 3% of that percenent
Yeah, so
have
actually, I didn't do
nonsense?
Yeah, I didn't do a very good job of explaining because I kept changing my number. But here's the numbers. So, 5%, about 5% of people with cystic fibrosis have 2 mutations that don't make protein. Okay, that's why you hear that last 5%. Those are people who just don't make protein, so we know they're not going to respond to typical modulators. There's 2 groups within that 5%. There's a group that have 2 stop mutations, would be one of those. There's another group that may have 2 of, there's other ways of not making protein. There's deletions. So people may have a deletion of a chromosome. There's also some very called canonical splices, which are basically where there's a problem with a gene that's not a stop mutation, but it keeps the protein from making it. So in general, about 2% have 2 of those canonical splices, and it's about 3%, maybe 2.5 to 3% that have 2 of the stop mutations. We'd have to look through the exact registry data because it depends a little bit by country, but I think in general you can think about 5% don't make protein, about half of that group is stop mutations, half of them with these other mutations, and there's a mix. Obviously, there's some people that might have a stop and a deletion, so that's another little bucket in there.
Are they taking nasal swabs on all 5%?
So that's a real good question. Right now they're focused on the stop mutations. And here's the reason why. It's specifically to be able to test therapies for the stop mutations. So if you have a deletion, it doesn't, for the current project, it doesn't help all that much. Really, in that group, what we're looking at other ways. We're looking at gene therapies, those type of things. We're specifically needed cells to be able to test things. Originally it was Adelorn, but other things which read through that gene. So we need them to have 2 copies of that gene. So that was very complicated. I'm sorry I put anybody to sleep there. All right. One last question? Otherwise, one last one.
Here you go.
Here
you asked that last 5 to 7% to go to the registration.
Oh, I'm sorry. That's perfect. Oh, thank you, Laura. Okay, here we go. So Laura reminded me that if you're in that last 5% of people who don't make any protein, or your loved ones are, or the 2% that have such rare mutation, or anybody, that group that feels like, hey, I'm going to need something special besides the modulators, there's going to be a meeting at 5 p.m. and a team picture, right? Oh, 5 p.m. That's actually right now. Oh, that's right. Yeah. Actually, it already happened, it sounds like. So follow Laura. She's headed, and it's going to be, where is it going to be, Laura? Registration. All right. Thanks, everybody. See you tonight.